Good morning to everyone. I'm Rajesh. I'm working at Children's Hospital, Boston, in Zone Laboratory. And today we'll be learning about new things about stem cells and how stem cells can be used to provide solutions to old issues such as sickle cell anemia and alpha thalassemia. Now, here is a red blood cell, which is full of lots of hemoglobin molecules, as shown here in the animation. A red blood cell is around 7 micron in size, and typically a red blood cell contains 270 million hemoglobin molecules. And that corresponds to around 30 picogram of hemoglobin, with each carrying around a 4 heme groups. The average adult human being manufactures around 2.5 million red blood cells per second. Adult hemoglobin, as shown here, contains 2 alpha and 2 beta chains, and each hemoglobin contains 4 porphyrin ring complexes. And thus, that allows it to carry 4 oxygen molecules. Now, by that logic, a single red blood cell can carry about 1.2 billion oxygen molecules. Now, each of these red blood cell develops in bone marrow in several stages. So it begins with a stem cell, which decides to make red blood cell, and the next cell after that is called as a hemocytoblast. And this is a multipotential cell in from the mesenchyme. Then this cell becomes erythroblast. Now in this case, this cell has already committed to red cell fate. In 2 to 5 days of development, this red erythroblast gradually fills up with lots of hemoglobin, as shown here in the animation. And this is what allows the cell to carry oxygen required for every cell in the body. Now, during this process, when it comes to human beings, hemoglobin is populated. It's made from the globin RNA. And to maximize the amount of the globin carried by this cell, human red blood cell actually throws away all the cellular organisms. So it throws out the nucleus, the mitochondria, and everything you can think of, which makes a cell a cell. And this really allows red blood cell to have lots of hemoglobin, and in turn allows it to carry a lot more oxygen. In the late stages of its development, the red cells are called as reticulocytes, which ultimately becomes fully mature and start circulating. And it takes around fraction of a minute for a red blood cell to go through the whole body and pass through the heart again. Now, red cell's job is essentially to carry oxygen from the lungs to the different tissues. And now you can imagine this tiny red cell, which is around 7 micron in size, really delivering oxygen to individual cells in your body for its metabolism. Now you will be wondering what exactly happens if the cell is throwing out all the required cytoplasmic organelles, including mitochondria. How does it produce ATP? So in case of red blood cell, there is a fermentation, which is a pathway separate from glycolysis, and a part of glycolysis, which involves fermentation process, is used by a red blood cell to make its own ATPs. Now, more than often, it so happens that when a red blood cell is going through different capillaries, as shown here, it can bend and really squeeze through tiny spaces. And its biconcave shape is something which allows it. Considering there is no nucleus, there are no other cytoplasm and specialized cytoskeleton, it can do that job and deliver oxygen. But once in a while, it just so happens that there is a mutation which is coding for hemoglobin gene. And it's just so happened that globin mutation leads to slightly faulty hemoglobin production. And in this case, we are talking about alpha thalassemia and sickle cell anemia like disorders. In this case, at least for when it comes to sickle cell anemia, there is a mutation which leads to bending and crescent shape formation of the red blood cells, as shown here right at the center. Now, these cells, in comparison to normal biconcave red blood cells, are actually very difficult to manage. And when they pass through these tiny spaces, they kind of block these the blood arteries. And this blockage of red blood cells can lead to severe symptoms. And many times it leads to infection in different organs and failure of those organs. Now you can also imagine that these red blood cells, which are crescent shaped in case of sickle cell anemia, are not the best cells when it comes to carrying oxygen. And this is something which leads to lots of different problems in the body, and the whole life experience of this particular individual who is suffering from sickle cell anemia is compromised. Now we know from our research and from the previous lecture that there are these specific stem cells known as hematopoietic stem cells, which gives rise to all the different kinds of cells in the body. Now, what we know from the previous literature and Till and Mackillop study that the stem cells are actually residing in the bone marrow, and they do have a capacity to form lots of cells, throughout the body, require as the body requires, and throughout the life of the animal. Now, what happens once in a while is stem cell gets a mutation, and it can be genetic, congenital, it can be something which can happen in the adult life. But in case of sickle cell anemia, what they have seen, there is a mutation which leads to changes right from the stem cell itself. And when the cell is forming different kinds of cells as shown here through the common lymphoid and common myeloid precursor, there are lots of cells which are compromised. And in case of sickle cell anemia, there's one particular path which is from hematopoietic stem cell to forming common myeloid precursor to the erythrocyte, which is the red cell, is compromised. Now, red cells, in comparison to all other cells, form close to almost 50 to 70% of the blood volume. And now you can imagine every cell in your body having this particular mutation, which makes it into crescent, a moon shaped cell also. And that creates a huge problem when it comes to, just normal functioning of the body. Now, the mutation I'm talking about is shown here, and we all know that DNA codes for RNA. So, whenever there is a normal gene, like as shown here, which codes for hemoglobin, it leads to a normal hemoglobin molecule as shown here. But in case of sickle cell anemia, what people have found is that molecule which is coding for normal hemoglobin undergoes a mutation, and now the GAG coding for glutamine, which is a negatively charged molecule and thus hydrophilic in nature, the changes to GUG coding for value. Now, in terms of amino acid properties, these two molecules are these two molecules are very different, and that changes the way the molecule really reacts when it comes to changes in the oxygen levels. So, for example, from being a normal hydrophilic molecule, it changes to hydrophobic molecule. And this one particular change in amino acid from being GLU to valine leads to changes in the whole architecture of the red blood cell. Now, you can imagine each of the red blood cell carries around 300 million molecules of hemoglobin, and every molecule having this kind of mutation changes the way the hemoglobin is organized inside the cell. Now, usually when the red blood cell has hemoglobin, it is soluble and globular, as you have seen in the animation. But the moment it takes the hydrophobic amino acid because of the mutation, the kind of actually starts aggregating and forms fibril inside the cell. And this nice bag of the hemoglobin carrying oxygen around now becomes a slightly tufted structure. And whenever that happens, and when it tries to go through the capillaries, it kind of ruptures. And that has been the problem in case of patients. Now, why we should study hemoglobin-related disorders when it comes to we as an Indian? Now, here is a world map, and as you can see, when it comes to yearly birth, there are so many number of mutations which happens when it comes to the birth, and there are close to 100,000 birth in India which have hemoglobin mutation leading to sickle cell disorder. Now, the burden of the people going through this sickle cell disorder is huge when it comes to Indian subcontinent, and hence it makes sense to study all these hemoglobin related disorder. Now, in addition to sickle cell anemia, what people have seen is alpha thalassemia, which is another disorder which leads to mutation in the alpha globin gene. And this one particular mutation leads to lower loading of alpha globin. Now, as I said, alpha and beta genes are actually a combination which forms the globin molecule. And this is what leads to low production of alpha globin. And having low amounts of globin per cell, considering each of the red blood cell carries around 300 million molecules of globin, you can understand the amount of the oxygen carried by the individuals carrying alpha thalassemia is also lower. Now, what people have found is when it comes to early developmental stages and the late developmental stages, as in the adult cases, there is a switch in terms of the kind of globins which are produced. In the beginning, the individual, when it's young, produces fetal globin, but then in the later stages, it switches to adult globin. And what they have found is fetal globin can actually carry a lot more oxygen and in comparison to adult globin. And that is something very interesting. Now, if an individual carries a mutation in the one of the adult globin, can you actually go back and switch on the fetal globin to cure the disease is something people are trying to understand. Okay, before I go for the actual gene therapy and how people at Children's Hospital and Dana-Farber are trying to circumvent this sickle cell disorder, I'll take some questions. Iqbal, you have a question? Go ahead.
Yeah, we see the in beta globin mutation, we see the glutamine amino acid transform into the valine and amino acid. Is it
Yes.
possible to valine or other amino acid will substitute there instead of glutamine? Why valine?
Why
This is my
valid? So
question.
what they have seen is when it comes to mutations, so the story goes like this: why one of the mutation is prevalent when it comes to human population? That's a question we are really asking. Now, some of these mutations, when it comes to beta globin gene, are also associated with. If you see the distribution, let me walk you back to the slide of the world map. And what they have seen, these the same countries where there is a high prevalence of this mutation, they found out there is a high prevalence of mosquito-borne diseases such as malaria. And what they found out is if you have this particular mutation in the globin gene, which leads to sort of sickling of the cell, if you have one single mutation, that gives you some protection against malaria. Now we all know that malaria sort of populates the red cell, and that's the favorite cell where it will make more number of parasites. What they found out, if you have this mutation which we just talked about, actually that gives you protection against the malarial parasite. And what has happened over a period of history, whosoever had that mutation, they survived malaria outbreak. Now you can imagine there are 100 individuals, some of them have malaria, and they happen to have this mutation. People who have the mutation in the red blood cell, coding for sickle cell disorder, they sort of survived. And the people who were carrying normal red blood cells, they died. And then the trend continued, and those were the people who reproduced and made more, individuals. And that something has selected these individuals over a period of time. Now, having said that, is the valine the only mutation which causes sickle cell disorder? Not really. There are other amino acids which are also leading cause of sickle cell disorder, but valine is one of the major sickling disorder. I mean, something which forms the major contributor when it comes to all the sickle cell patients. Yeah.
So you say this mutation occurs because of to create a protection against the malaria disease or malaria
So the
prevalence.
way the mutation occurs, actually it doesn't occur to protect against it. It just happens randomly, and it happens randomly, but you're selected because of the external pressure. It's like environment. Environment has certain things going on irrespective of what you're thinking. But it just so happens that you have certain capacity. Like for example, if you live in Savannah or South Africa, if you're not a good runner, good luck. You're getting it that simple as that in the primitive times. Somebody's going to chase you down. So, and it just so happens, you are slightly better at running than others, and then it just happens that you have better muscles, or you're fed nicely, and that kind of selects you over other individuals. So it's a natural process, and that's how evolution works. There is nothing very directed till now, until now. Yeah, now the selection can be very, artificial. Like for example, when people are trying to edit out genomes, the selection can be very artificial, and at the same time, humans can be considered as a, agent of for nature. So, but most of the evolution happens without really, preconceived notion. It's just selection happening at every place. Yeah.
Rajesh, I also have one question.
Oh, please shoot.
This is sickle cell anemia. Are all the RBC cells or sickle cell anemic, or only few?
So in case of an individual who have mutation in sickle cell disorder, what happens is it's a genetic mutation and which happens at the right at the birth. So every cell in your body is actually a potential sickle cell in a sense. But then the shape change happens in based on the situation. And so you can have all the cells which are capable of sickling, but when it comes to complications, it's only few cells which sickle based on the local oxygen exchange and the group of cells, and that is good enough itself to really cause problems. So most of these individuals, when they have these problems, they have to visit hospitals every 6 months for transfusion, which essentially means taking out all your body blood and then replacing with the red blood cells, because red blood cells have a half life of close to 120 days, and by then the newly fed red blood cells are already out of commission. They are destroyed in by macrophages and all to make place for new cells, and so you need to remove the damaged cells. At the same time, you are producing sickle cell red blood cells. So you need to visit hospital, and it's a very painful process and very costly as well. Yeah.
So there is we are now using gene expression. So why can't we remove that gene and introduce the normal gene which produces the normal
Awesome.
RBC
So
cells?
I'll come to that in a minute. We are just going towards it. Yeah. Iqbal stopped me for questions, so we are getting there. That's a very good question. Yeah, very good thought. Yeah. Who's
Okay,
next?
Rajesh.
Me, Rajesh.
Yeah, Akhila, go ahead.
Yeah, how stem cells are destined to form different types of cells, like two progenitors form different types of cells. How a particular progenitor knows that it need to form such type of cells?
Yeah, that's a very good question. So, that has been a question which people in my field at Children's Hospital Boston and many different stem cell systems have been trying to understand. What they have found is when it comes to a stem cell, the chromatin, the DNA is really open, and it has all the promoters, which are the driving forces behind every gene, open. Now, the only time a cell becomes red cell is when the module for red cell development is activated. And the activation requires transcription factor and RNA polymerase II to bind to those promoter regions. Okay. Now, the signaling for the transcription factors to bind to that particular region comes from outside, and that's where the niche comes in. And the niche, now you can imagine, has lots of different kinds of cells, and now you will ask, how do the niche cells know what sort of signal to provide? Okay, now just imagine if you go for a run and you really need lots of oxygen and you do that on daily basis, then your body is asking for more oxygen, and that is something which is sensed by your whole body. And the niche cells, the niche cells are telling stem cells that your body needs more oxygen, we need more red blood cells, so produce more red blood cells. So niche cell will tell the stem cell that this body needs more red cells, why don't you produce more stem cells, more red cells. So, and then the stem cell will get activated, it will come out of its quiescence, and then it will go to common precursor which is involved in formation of red cell. Now this process is really complicated. Now you can imagine a stem cell has its own set of mRNAs. A red cell has its own set of mRNAs, which means you need to get rid of the stem cell related mRNA from red cell related mRNA and make protein out of it to form fully mature red cell. And that is something we will be talking about in fourth lecture. Who's next?
Rajesh, one more.
Yeah, please. Sure. Sorry. My apologies.
It's okay. Red blood cell initially contains different type of organelles, right? Especially nucleus.
Yeah.
So the later stage of its life cycle, it gets removed.
Yeah.
So where does that nucleus or the different cell organelles go?
Very good
Does they are
question.
removed from the body?
Yeah, that's a very good question. So, when it comes to considering we are producing close to 2.5 million red blood cells every second. So just imagine the amount of the material it has to get rid of. In a normal human body, one red blood cell production takes around 2 to 5 days. And it's a machinery like a conveyor belt, in manufacturing industry. Now, the amount of the waste generated, which has a nucleus, mitochondria, and other cell organelles, is huge. So, body cannot simply just throw it out. So, lots of these things are recycled back, and macrophages and other cell types are really important when it comes to this process. So, what happens is whenever a cell wants to give out the cellular organelles, these other cells of the body, such as macrophages, as I said, will take up that, and then they will chew it out and will be used for other metabolic activities. So nothing is actually wasted in some sense. And spleen and other places are known to be major when it comes to this activity, spleen, liver, and other places. So all the cell organelles will be put to good use, and it will be energy efficient process. Good question.
Thank you, Rajesh.
No problem. Next.
Hi, I had a couple of questions.
Yes, sure.
So, when you talked about sickle cell anemia and there being like this mutation in the gene which causes it to be valent and hydrophobic, so can't we just use like CRISPR to like fix it? And
Yeah. So CRISPR is a very interesting technology. For those who don't know, what CRISPR has has an enzyme known as Cas9, and that gets a guide molecule which is RNA. Now, this guide molecules, along with this nuclease, forms a one-type couple, and this is something which serves as a scanning machinery throughout the genome. Now, based on the sequence of the RNA, you can really go specifically to a location where you want to edit and really create a mutation of your choice. What people have found when they were trying to standardize this technique to use and make very specific mutation, that this whole process of finding a specific gene locus and really editing the gene locus you are interested in was not sufficient enough. And then it would also go to site A where you want to edit, but also will go to site B, site C, where there is some sequence similarity, and really edit those regions as well. Now that is a big problem. If you do that with the stem cell, which is capable of giving rise to lots of cells, you can just introduce one mutation, which will give you cancer. And so it took some amount of standardization. And yes, at this point, people can use it, but you can imagine it takes a lot of efforts to find out the correct sequence and correct, editing. And a lot of times, when you introduce a Cas9 and a guide RNA complex, that's what it is called, RNP, ribonucleoprotein inside the cell, the editing efficiency is not 100%. So if you take 100 cells and if you introduce into 100 cells, only maybe 10% of the cells will be edited to your satisfaction, and it's a slightly low efficiency process, but it does work, and that's why the Nobel Prize. Yeah, good question.
Right. Okay. And what about using ZNF to remove the mutation and some DNA ligase to put the proper nucleotide? Is that possible, or is it only CRISPR?
Actually, in comparison to the zinc finger nucleases, CRISPR-Cas9 is considered as a far efficient, better technique. People are, I think, there are very few people who are using zinc finger nuclease at this point. It's slightly less efficient process. What you can do with CRISPR-Cas9 and editing technology is, when a cell is being edited using CRISPR-Cas9, which removes, cuts two strands of DNA and removes a chunk of the DNA, you can provide cell sequence of your choice. And in that sequence of your choice will be used as a Xerox copy machine to make a double-stranded DNA, and that will be edited into the cell. So you can literally remove a good piece and replace it with another piece which has altogether different sequence, and that's possible. So people are trying to do that. We are actually trying to do that in my own project, and I'll talk to you about that. Yeah, that's a very good question.
Okay, great. And also, you talked about a fetal globin having more oxygen
Yes.
instead of adult globin. So, is there some because it is still not mature, is that the reason why it still holds more oxygen, or is there some other factors which factor in for it to hold more oxygen, and why is it not conserved as it moves into adult globin?
So what they have seen in terms of the fetal globin switching to adult globin, the if you give to clarify what exactly what I meant when it can carry more oxygen is, if you give one molecule of oxygen to a fetal hemoglobin and to adult hemoglobin, then every time when there is a competition, fetal globin will scavenge that oxygen, which means the binding affinity for the oxygen is higher, which means that it will carry the same amount of oxygen as the adult globin, but it will carry it with higher efficiency because it has higher binding. Now, what has happened with the time, the fetal globin has that capacity because when it comes to development of a baby inside mother's womb, it has to take that oxygen from mother. So red blood cell of the mother will be carrying oxygen, and now the baby's globin has to take it from the mother so that it can support its own growth. As simple as that. Now you need a competitive binding, and in that case, the fetal globin really does a great job. Now, once that happens, that requirement is done, the baby is born, and in the adult case, you don't need such a globin, which will be very strong, and that changes to from the fetal globin to adult globin. And you will see how the globin genes are organized on the locus. They are arranged one after another, and it's just amazing to see how they are regulated at promoter level and how the globin switch, that's a term, happens. Yeah, I'll come to that in a minute.
All right,
Next.
thank you, Rajesh.
Awesome.
Yeah, I have a question.
Yeah, I just wanted to correct myself. It is glutamate, not glutamine. Okay, so for your information, it's a negatively charged amino acid, not the positively charged amino acid, which is glutamine. It's a glutamate. Yeah. Sorry, Ashini, go ahead.
Yeah, that's fine. Like, is there any possibility or a case where the stem cells produce normal RBCs and then later it will produce a sickle cell anemia, or it is being inherited only like from the birth only is possible, or it can happen? Excuse me.
In the
My
later
voice.
stages. So
Yeah.
what happens is, till now what people are seeing is sickle cell anemia is something which is a genetic trait. It happens right at the birth. But having said that, uh, the quantum loading of the globin for each red blood cell is something which is slightly variant when it comes to different individuals. There are some individuals where the amount of the globin is low, and that's what causes the thalassemia disorder. Now, people have seen there are mutations which are supposed to cause thalassemia, and in that case, they don't get thalassemia. So there is that mystery. So it can happen that there are two individuals have the same mutation and still do not have red blood cell disorder. And it's a much complicated research area. We are trying to understand what exactly happens. Now, a lot of times when you say a sickle cell disorder, it's a single gene related disorder. There is no complex set of network involved. Like there's only one gene, right? It's a single one, what they call as a unigene genic disorder. But when I say there is a mutation and still you don't get thalassemia disorder, that raises a question, which means there might be another gene involved in that process. And what we are trying to find out actually in my studies is probably when it comes to RNA, besides having mutation in those nucleosides, there might be some modification on top of it, which is added by another gene. And if you have mutation in that gene, which is involved in addition of these chemical nucleotides, chemical moieties on the RNA, then probably you might develop a red cell disorder. It's very complicated to explain at this point. Maybe in the fourth lecture it will be clarified. Ask me again in the fourth lecture. Yeah.
Yeah, okay. And like when we talk about these carriers where both normal RBCs are produced and crescent shape RBCs are produced,
Yeah.
in those cases, like when the RBCs have a total lifespan of 120 days and when they are destroyed and again they are being produced, so how will that cell know, like, is it like some stem cells are being destined to produce these crescent shaped cells and some produce normal RBC cell? Like, how is that mechanism happening in case
So
of
what
carriers?
happens is when the cell is being born from the stem cells, all the stem cells in this patient's body have the mutation. So every cell being produced is a potential sickle cell. Getting it? The only time it will undergo sickling is when the situation arises, there is oxygen exchange, and that causes the conformational change. Okay. So every red blood cell in this person's body can undergo sickling. It's just a matter of time when it will sickle. So all the cells are identical. The shape change is something which happens only when you ask it to do a job. It's a lot like flexing a muscle. Your muscle is always there, but only time when you are asked to lift something, you can see if flexing it and then bicep coming out or tricep coming out. It's that simple as that.
Something like oncogenes, when they are activated, they turn into cancer cells.
So oncogene is slightly different process. So what happens in case of oncogene is a cell is doing its job and then you get a mutation and then that goes rogue. But when it comes to production of red blood cell, it is something which is happening normally. The only problem is the building block itself is compromised. So it's not a special activation, but when a cell is fully functional and when it is trying to do its job is when there is a problem. It's really like when it comes to application of something, if the part is faulty, that's the only time when you know it's not going to work. As simple as that.
Okay,
Yeah.
okay, Rajesh. Thank you.
Awesome. Thank you. Next.
Hello Rajesh.
Hi.
I just wanted to ask, Yashini asked my question actually, which I had in my mind. So
No
yes,
issues.
I
You can
will.
ask again.
Yeah, no, it's fine. I heard the answer,
Okay.
and
I'll give you a different answer.
okay, maybe later.
Okay.
I wanted to ask that a person having sickle cell anemia, can he or she have that? Can he or she undergo leukemia or suffer from leukemia?
Yeah, that's a very good question. So what they have seen whenever it comes to sickle cell disorder, um, sadly, everything about your body function and physiology is compromised. And that many times leads to other complications. So there is a good chance that you might get another mutation and then you will develop leukemia, which will be slightly worse than other patients. So there is a good chance. Now, the reason why that could happen is because oxygen is very important when it comes to maintenance of niche and in general body physiology. And any change in the niche can lead to aberrant changes in terms of epigenetic modifiers. Now, having said that, have they actually studied if a sickle cell disorder human being gets more leukemia or not? I'm not sure at this point. I'll read upon it. Yeah.
Okay, and I have another question. Actually,
Please.
when Rajan was taking his class, I asked Rajan, but Rajan said to ask you. Okay.
Yes.
So I asked that do RBCs undergo autophagy?
So what they've seen is, red blood cell, if you say, only the mature red blood cell does not have the necessary organelles and all. But then it is known to undergo certain membrane related processes. So they may or may not do actual autophagy, but what people found out that they have these receptors on their membrane, which are now what, there is a very cool study that RBC is not only there to carry oxygen, it also senses danger, like, DNA, which is not supposedly should be there in the cytoplasm, in the blood. So they have these receptors on the membrane which can bind to this extraneous DNA and reactivate other immune cells. So they may or may not have actual autophagy because it requires lots of membranes and all, but they have some residual activity going on, which may or may not be autophagy related. Now you would wonder, why do the red cell will require autophagy? So autophagy is required for cellular organelle maintenance and all. So if you imagine, when the maturation of red cell is happening, there is a huge amount of all of these activities. Every organelle in your body has to go. So you can say that till it is being matured, it has maybe highest activity of autophagy. And then it gets rid of everything, every part of nucleus and every cytoplasmic molecule and an organelle not required for globin carrying. So yeah, I would say yes, there is autophagy till very long time, and then there is probably very slim or none activity. Yeah, very good question.
Okay. So
One more thing I want to add to that
yes,
is,
sure.
having said that, we are just talking about human red cells. What about zebrafish in dogs and other animals?
Okay, yeah.
They have nucleus. Their red blood cells have still nucleus, so everything goes fine in those cells. Yeah.
Rajesh, sorry, Sumay.
Okay. So basically, till the terminal differentiation of the RBCs, like till that time, they undergo autophagy. But after that, they go like they can opt for apoptosis or programmed cell death, right?
The cell death is something which happens only when a cell is not required. If you require a cell, then there is no apoptosis.
Okay.
Yeah, so
Thanks.
you need a red cell. Why would you introduce apoptosis? Only when the differentiation process, which has to go smoothly, if there is a problem, then the cell has to undergo apoptosis. Otherwise, everything is fine.
Thanks.
No problem.
Rajesh, Rajan also told me to ask this RBC related question to you.
Yep.
So I asked him that RBCs have a very short lifespan. So like comparatively to other cells. So is it because of the fact that there is no cell recycling mechanism like autophagy,
Yes.
or is it because of the oxidation? I don't know. Is it because of that, or what is the reason?
So what happens is, that's a very good question. Why do red blood cell has to be destroyed at the end of 120 days? And it undergoes lots of these, gargantuan number of cycles when it comes to oxygen exchange. And each of the, as I said, each of the red blood cell has around 2.5 million hemoglobin molecule that effectively corresponds to 1.2 billion oxygen molecules. Now you can see, imagine the cell is exchanging oxygen and carbon dioxide at that rate, and that introduces lots of damages to the globin molecule and to the membrane. And considering there is no mechanism in place to repair it, it has to be destroyed because it will be a nuisance after a while. So it is destroyed after a while. Yeah. Next,
All right,
Manusha.
thank you so much.
Yes.
Awesome.
I was just thinking that if we cannot change the sickle cells, can we have some drug which will make the oxygen the oxygen will be okay with the abnormal cells, like it will behave, the oxygen will behave it will see the sickle cell as a proper cell?
Yeah. So what people have been doing, and so far it has shown to work, is hydroxyurea, which prevents sickling of the red blood cells, and that is something have been used in many cases, but it may or may not necessarily work in everybody's case. So yeah,
Okay.
yeah. Okay. Next, Akila, you have another question?
Yeah.
Yes, please.
Some mutations are possible in WBC that alter the shape of WBC, and WBC are mostly involved in immune responses, and they
Yes.
play important role in maintaining immune system, right?
Correct.
So does any mutation happens in WBC that they alter the shape, and how does it affect the immune mechanism inside the body of an organism?
Yeah. So what has seen when it comes to a white blood corpuscles, there are lots of different kinds of cytoskeletal elements which are involved in development. There's one very peculiar gene. You might want to write it down. It's known as piezo, and it is something people have seen involved in lots of stem cell maintenance and other processes. And mutations in this gene can lead to bone marrow failure and, in general, problems when it comes to stem cell development. So yes, there are a lot of other mutations when it comes to cytoskeletal elements involved in the functioning of the white blood cells. Uh, when it comes to cytoskeletal elements and leading to shape changes, in general required for migration and other processes, mutations like those not necessarily will only change white blood cells. It will also change every place wherever cytoskeletal element is required. Like for example, actin, myosin, and there are other the molecules which are involved in the actual migration of the process cell. And if you have any change in those uh, uh, molecules, there is a lot of problem. Ron Whale, who studied all these processes for all his life, has amazing laboratory. You might want to check out his research. You will be surprised what all can change if you have mutations in myosin and other cytoskeletal elements. Yeah, good question. Yes, Chamundeshwari.
Thank you, Rajesh.
Yes, Rajesh. Like previously, Sharvar, like while discussing with Simoy, you have
Yeah.
told that the organisms like camel and lavus have nucleus, right?
Yeah,
In
zebrafish. I would say zebrafish. Let's stick to zebrafish. Yeah.
Okay, then see, have you like anybody tried or experimented to like change the glutamate into valine and see the mutation? And because in humans, it has no nucleus. So is it the reason that it is like constricted so much and becoming a sickle? Like when you like try to induce the same mutation in other organisms, like in zebra, is it the same sickle shape? Because it has nucleus, what shape would it undergo if we
Yeah.
induce the same in
So you're really asking what is if the quantum loading of sickling globin, if change, can it lead to different outputs? That's a question. That in a cell, if you have lower amounts of globin, will it lead to sickling or not? So that is something I'm going to come to. So stay tuned for it. Okay, just to give you an answer, if you have a mutation which is causing sickling of the globin, no matter whether you have a nucleus or not, it will cause problems to the cell, because once the sickling starts, which essentially makes a soluble protein which was carrying oxygen into a fiber, and that fiber is something which can happen even in the presence of nucleus and other cellular organs. And once that fiber happens, that sort of pokes into other membrane such as the cell membrane and ruptures the cell and causes the sickling and lots of different things. So presence of nucleus may not change much, but if you just change the amount of the globin which is causing sickling, and if you supply it with the normal globin, that can have an effect. And I'll come to that in a minute. Okay, so I'll move on.
Okay.
Very good question, guys. Very impressive. So then, as you all pondered upon, that if there is a mutation can we just not fix the mutation? And in this case, there is one particular way of doing it. As you said, Sharwari and others, that if there is a mutated globin, can we just replace the mutated globin using any other way, such as lentiviral vector or CRISPR-Cas9 engineering? And people have been trying to do that. So in this process, as shown here, a mutated globin, as we discussed, leads to a sickling of cells, and that takes a crescent shape. And whenever there is a gene mutation, as I said, it leads to this fiber formation. Ideally, it should not have happened. Now, if you want to change that gene which is causing sickling, it has to be done at the stem cell. And then in that case, you need to use these vectors called as a lentiviral vector. So what these viruses are essentially packages which are modified from actual viruses. So they do not have genes for causing viral disease, but they have everything we want to put in our stem cells. So scientists around the globe, they take these lentiviral vectors and then they add the gene they want to edit inside this cell. Now here we can imagine it is carrying RNA for coding a normal beta globin gene, and that lentivirus is then introduced into a stem cell which can form lots of red blood cells. Now in this case, you can imagine a red cell coming out of this stem cell which already had one mutated version of beta globin, and now they are supplementing another beta globin version which is a normal globin. And in this case, the production of the new beta globin will be something which will help avoid the sickling of cells. So there will be a lot more amount of normal globin which is produced in comparison to the sickling beta globin, and that's why the cell will be normal as shown here. But it will still have some amount of sickling causing beta globin, and that's one of the technology which people have been using. Now you can understand this is not really the best way of going about the problem. Now for this particular experiment, the stem cells come from this particular niche known as a bone marrow of human being. They are seen in case of pelvic girdle and the long bones of the body such as femur, and they are situated in these very tiny pockets as shown here in the cross section inside the bone marrow cavity. And it's a very specific niche, and to identify such a stem cell which corresponds to actual multipotent precursor is very hard, and people have found out different markers on the stem cells. So when you want to do a gene therapy, as I described, you need to take out some of these bone marrow stem cells by giving GCSF, and that allows the proliferation of stem cells, and some of these cells are released into the blood circulation. Now scientists take out those blood cells and the stem cells and then really do the lentiviral vector introduction of this normal globin, and then the new stem cells which have extra copy of beta globin is reintroduced inside the animal. And in this case, a patient suffering from sickle cell disorder. Now coming back to when it comes to globin production, as shown here in this graph, which is slightly complicated to understand, but we'll go through it slowly, is when it comes to early gestation period, as early as few months into development, there is a huge amount of production of different kinds of globin, and in this case it's shown as epsilon chain, which is made along with other alpha globin. In the later stages, this particular globin is replaced by gamma globin, and this is something which is called as a fetal globin. And whenever that change happens, you can see the development of blood changes from yolk sac to the liver to the spleen, and eventually in the adult, in case of healthy individuals, in the bone marrow. Now, when that transition happens, the amount of the gamma globin which is produced significantly decreases, and then the beta globin chain is put to action. Now, when we see the arrangement of these genes coding for different globins onto the chromosome 11, where the region codes for all the globins involved in a hemoglobin production are arranged in one line in the same sequence as they're expressed during development, and that's very cool. Now, the region right before the globin is called as the locus control region, and this is something which people in the field have found out are the promoters required for the expression of these genes. So every time a new globin is produced, a different locus control region is put into action, and that's how the production happens. Now, whenever there is a switch from the gamma globin to beta globin, the locus for controlling the gamma globin is shut down, and the locus controlling for the beta globin is switched on. And this is all happening at the DNA level when it comes to mRNA production. Now you will ask why does how this particular process happens and how the switch from the gamma globin, which is a fetal globin, to adult globin happens. My colleagues at Hematology Oncology, Stuart Orkin, Vijay Shankaran, and Nan Liu, found out there is one particular protein known as the B cell lymphoma 11 or BCL11A, which binds to this one particular locus and shuts off production of the fetal globin, as shown here in the schematic. And that is a signal for activation of beta globin. Now, if you have a normal copy of beta globin gene, then you will have normal blood cells. But if you have a mutated copy of beta globin gene, then you will end up having sickling of the cells. And in that case, it will be nice if you can just continue to express gamma globin, leading to fetal globin production and leading to a normal red blood cells. And this is something people are trying to explore as a therapy. So in nutshell, what Vijay Shankaran and Stuart Orkins and David Williams at Hematology Oncology and some people at Dana-Farber are trying to do is, by taking those stem cells which have mutated copy of beta globin, can you actually repress expression of BCL11A, which was actually suppressing the fetal globin production? And people have been doing that successfully using mouse models and using cell lines. And much recently, there have been clinical trials in collaboration with Dana-Farber and Bluebird Bio company in Boston. So the therapy goes like this. You have a BCL11A protein, which was suppressing expression of gamma globin. All you need to do is remove BCL11A from the cell expression. And BCL11A is something which happens is expressed in the adult cells. Now, all you need to do is take a CRISPR-Cas9 technology and really knock out BCL11A, and thus allowing repression of, taking away repression of the fetal globin. Now, the moment fetal globin expression goes up, interestingly, the beta globin expression goes down. So you have a dual effect of production of a nice new fetal globin, and the mutated copy expression goes down. And this is something which my colleagues here at Children's Hospital have been trying to do. Now, there are many different problems with when using CRISPR-Cas9 technology, and what people have found out, you can use shRNA and really use the lentiviral vector, which I just described, and just knock down BCL11A at the mRNA level. And this allows you to take down BCL11A protein at the same time not introduce any genetic mutation inside the stem cell, which can cause different problems. Now, here is a patient. His name is Manny Johnson or Emmanuel Johnson, and the doctor who treating him, Erika Ezrick, who is a senior colleague of mine at Dana-Farber. And they're spearheading this particular clinical trial. And he's the first recipient of such a technology where the shRNA was delivered into his stem cells. So the stem cells from Manny was taken out. He was suffering from sickle cell disorder. His stem cells were taken out. They were reintroduced with this lentiviral, which is expressing shRNA against BCL11A. And they found out when they reintroduced these stem cells, which are producing now fetal globin, he used to require blood transfusion every 6 months. And last year or so, he has been off these blood transfusions and thus curing the disease. And hats off to Erika Ezrick and David Williams and Bluebird Bio team to come up with a technology like that. And at this point, Manny has a brother whose name is Aiden and who's also suffering from similar mutation. Now you can imagine there's a mutation in one of the parent which is causing the sickle cell disorder. And from the global map we have seen, besides India, it's actually African continent which is very big when it comes to sickle cell disorder. And because of the global migration, the number of patients when it comes to America are also increasing. And that's why there is a lot more research going on when it comes to sickle cell disorder and other red blood cell disorder in United States. With that, I would like to stop and take questions. If I see there are a few people who have questions. Manisha, go ahead.
Yes, I was just thinking that instead of the gene, can we just use the proteolytic pathway for like the BCL11A? Can we just throw it into a proteolytic pathway?
That's a very good approach. So, you know, it raises a question. You have a technology that works, and why cannot it be commercialized and made available to everybody in India? Because there are 100,000 kids born every year. Now, if we back calculate the amount of the money it takes for anybody to take out stem cells and reintroduce a new gene copy and really do the bone marrow transplantation, the cost is huge. And that sort of demands, what Manisha was referring to, a proteolytic pathway activation to specifically target only BCL11A and target its destruction. And so people are trying to do that at this point because gene therapy is a non-viable option when it comes to a mass of that size. So there will be a day, one can possibly use proteolytic pathway to really take down only BCL11A and thus really restore the levels of fetal globin. Very good approach. Very good.
One more thing, we know about we have heterochromatin and euchromatin, right?
Yes.
So if we can make the region of the BCL11A gene into a heterochromatin,
Yeah.
then I think it is one more approach to solving the
Yes.
problem.
So there are many different labs working on different angles when it comes to BCL11A, and introducing heterochromatin in BCL11A locus is something very cool. Having said that, BCL11A has other functions when it comes to other cell types in the
That
red
is
blood,
so.
in the blood development, and people are trying to circumvent somehow. And what they have been doing is, rather than using a stem cell specific promoter, they are trying to use a much specific promoter when it comes to red cell development. So now we can imagine.
We can use um, like a promoter which will be absolutely specific to the red blood cells.
Yes, that's what
Tissue.
people are trying to do. Yes.
Yes.
That's a very good thought process. Very good. Proud of you. Next.
Thank you.
Who wants to go next? Sharwari, do you want to shoot?
Yeah, sure, why not? Okay, so I had a couple of questions. So you said that the mutated copy and the fetal globin production is inversely, like if one is producing, the second one
Yes.
is inhibited. So why is there like this sort of like is it because of the detection, like like I'm not saying that they have feedback mechanism, but it's sort of like that, right? Like one inhibits the other. So is it because of that, or is it just some other mechanism altogether?
Yeah, that's a very good question. So a lot of work from Vijay Shankaran and Nan Liu and Victoria and Stu's lab, Stu Orkin, who's a senior colleague at hematology oncology. Uh, uh, what they found out is the locus control region as shown here. It's something which is inhibited from binding to the promoter region when it comes to gamma globin. And the binding is something which involves bending of the chromatin. So the moment BCL11 binds to that region, the binding which will bring this locus control region proximal to the promoter for gamma globin is somehow hindered. And that is something known to inhibit the expression. So the moment you start expression for the other gene, there is a sterical hindrance for binding of RNA polymerase 2 because the BCL11A or the promoter required for that particular gene is avoided. So it's not at the globin level which is being regulated. It's actually the genomic level where the regulation is involved. And I'll send you some papers to read about. It's a very cool question. It took almost 15 to 20 years for people to figure out what exactly was happening. It was a very good paper by Nan Liu, who's a friend of mine. who published a Cell paper and then Nature Genetics much recently. Yeah, prolific researcher. Yeah.
Okay, and one more question was regarding the BCL11A locus that we have. So in the locus, you mentioned that they are present in the same way in which they are expressed. Like basically, the
Yes,
first one is expressed
that
first.
is a very cool thing.
Yes, exactly. So I wanted to ask that is there a reliance of so suppose this so suppose like this factor A gets like expressed first and then factor B gets expressed second. So if there is faulty expression of factor A, does that affect expression of factor B, as in is the um proper expression of factor A necessary to activate the expression of factor B, proper expression of factor B?
So when it comes to locus control regions as shown here, if there is a problem in terms of any of this locus control region, the next locus control region is somehow put into action. Now the mechanisms behind that are not really known. Having said that, in case of zebrafish, there are a lot more number of globin genes and they have similar locus control region. So it's an area of active research how at genomic level, the expression of different molecules is controlled. Yeah. Sorry, I cannot give a satisfactory answer because the research is
No,
going.
no, it's all right. No problem. Thank you so much. And as Manisha talked about there being a promoter, like specific
Yes.
promoter. So why is it so, I will not say difficult, but tedious to get a promoter that is specific?
Yeah.
Because there are countless promoters and countless like locuses and pathways, or like, that have promoters on them. So
Yeah.
is it, why is it so difficult? And also as you talked about this LCR promoter being on this locus, so
Yeah.
can we just modify this promoter to get a specific promoter, or or what?
Got it. So what they have found, this raises the question of how any gene in a cell is actually regulated at the in expression level. So it was a very interesting study by a colleague of mine at NCBS, and he was looking at V-glutamate, which is vesicular glutamate, gene involved in various neuronal processes. Glutamate is a neurotransmitter, as you all know. What he found out when he took a upstream region of this gene, the not the actual coding region, but the gene region which is just up in upstream of this coding region, it was like several kilo base pairs of this region which constituted promoter, enhancers, and all the different elements. And when he took, rather than taking the kilo base pair, when he took 100 base pairs and 200 base pairs and such regions, he found out that and used those regions right in front of a GFP coding sequence. And when he put them back into the Drosophila, what he found out, every tiny sequence was now lighting up a different neuron. And when that result came out, we were very dumbfounded that we just realized that every small region codes for a very specific signal for very specific neuron to activate the same gene. What does that tell about the complexity? So the complexity of this region is such a huge that when it comes to its expression, it is expressed in lots of neurons, but every neuron is using different region for its expression. And that is the problem when it comes to using promoters. We have a level of understanding to say that, oh, this is a promoter, but we cannot say that for sure. If I take this promoter, it will express only in this one particular cell. Now, the problem is you have a sequence, but when it comes to expression of genes, it's not only that sequence. Locus control region is several kilo base pairs away from actual gene. What it does, it loops onto it, and then the regulation is happening. Now just imagine, how do you explain something like that? If you want to come up with a gene therapy, you have a promoter and you introduce into the whole genome, you don't know where it's going to land and how it's going to get controlled. But people are trying to get to that place. They're inching close. There are these synthetic promoters being made. There are mutations introduced to make it very specific. And at this point, we do have some understanding. So for example, when it comes to zebrafish, we use this promoter called draculin, which is expressed only in case of certain red cells and blood cells. So, and we have some understanding that it is expressed by a certain way, but still it is not the best promoter, I must say. Yeah.
Okay, that's very
No problem.
interesting.
Very good
And
question.
that, and my last question was that you in sickle cell anemia, the mutation causes the amino acid to be valine, which is hydrophobic,
Yes.
and that is the whole problem, that it's hydrophobic and it causes conformational changes. But what if the mutation was still a hydrophilic amino acid, but not the actual glutamate? I'm sorry, am I right? Glutamate, what
Glutamate,
was
yes,
the...
correct.
Yes. So what if the mutation was not glutamate but another hydrophilic amino acid only? So I know that there will be some changes in the many changes in the functionality, but would that also induce like such a threatening situation on the RBCs and induce a conformational change?
So what they have seen when a amino acid is replaced by another amino acid of similar chemical properties, the function is compromised, as you said, but it is not completely abolished, and so the defects are less. But what people are trying to also understand, coming back to the chemical modifications on RNA, every molecule, every amino acid is coded by certain codons and anticodons, and then there are certain tRNA molecules which will bring in those molecules. And what people have seen, what we understand from the tRNA is not all the tRNAs are expressed at the same level. There is codon bias, and a lot of times those codon bias can dramatically change how much of the protein has to be produced in that given time. Now, as I said 3 times already, every second we are producing 2.5 million red blood cells. Now just imagine your efficiency going down by 1% can lead to dramatic reductions in terms of how much red blood cell of good quality you can produce. So it will have an effect. Okay, Vaishnavi,
Thank you, Rajesh.
awesome questions. Very good. I'm really proud of everyone. Yeah.
Yeah, I have one question that if due to some mutation in our RNA, a base pair which should be guanine, instead it was replaced by adenine, but our nitrogen bases could undergo tautomerism. So the immune form of adenine would resemble guanine. So if our RNA could detect this mutation and means tautomerism and make it into immune form, and so that tRNA could read it as the same codon and place the original amino acid, whether this kind of repairing mechanism possible in our body?
Yeah, so a very good question. So what they have been doing recently, they have found out a lot of RNA editing enzymes. And what they essentially do, as you said, they edit the nucleosides at the RNA level, not the DNA level. And ADA is one such enzyme I'm working on. It's known as adenosine deaminase. And you can indeed change A to C and then eventually code for altogether, different amino acid. And indeed, it can change the way the tRNA will probably come and recognize that amino acid. So it is a viable therapy. Very good thinking. Awesome. Akira?
Yeah. As we know that liver in the at the fetal stage act as hematopoietic organ, right?
Yes.
In the later adult stage, it loses the capacity, and only bone marrow is responsible for production of RBC, right? So, what
Yes.
makes liver to lose that capacity?
That's a very good question. Interestingly, we in Zon Lab have a project on it. I'll get detour from the question and come back to the question. Sorry for this. So what they have found out is whenever you're doing a bone marrow transplant in a patient, you're wiping out their bone marrow by using radiation. And this is a time when there is no niche for new stem cells. And what Len thought, and a colleague of mine, Tian Shah Han, who's a very good friend of mine, they thought that can we make another niche in the body, and they chose liver for it. And as you said, yolk sac, liver are the initial stages where, organs where the hematopoiesis happens. So they said, can we make liver into a hematopoietic organ and make stem cells rest there when we are doing all this interesting chemotherapy and introducing new bone marrow, so that there is high recovery in general when it comes to stem cells. Now, what happens whenever the organ develops, there are certain transcriptional network which are activated. And those transcriptional network activation causes some of these the niche cells which were there to take up a different job are actually die out in the process. And people are trying to understand what exactly changes in those cells. A colleague of mine who just started his lab in Boston University, Elliot Hagedorn, he was looking at, when it comes to zebrafish as shown here, the tail region of zebrafish actually is the place where in the early stages of development, hematopoietic stem cells are found. And as the animal progresses, the stem cells from the tail region migrate into something close to the heart and forms a new home. And this particular part is called as a kidney marrow, which is very similar to bone marrow. Now you ask the question, what exactly happens to this particular part? In case of humans, it is liver and yolk sac. In case of zebrafish, it's known as CHT, caudal hematopoietic tissue, CHT. And that is essentially destroyed in the process. We don't know if all the cells are destroyed or only some of the cells are destroyed. Do some of the cells migrate from that tissue to go into the kidney marrow is a $1 million question. And my lab is actually, Zon Lab is actually actively working on it. So stay tuned. Keep searching for Zon Lab publications and you will see something very exciting. Yeah.
Thank you, Rajesh. One more question.
Oh yeah, sure. Go ahead.
If a fetus which is inside the mother womb, can we check for the genes that are responsible for sickle cell?
Yeah. So what people have been doing in the early pregnancies, and something you guys will eventually figure out a better way of doing it. I am very confident of some of you will come up with a better technology. Is they take a needle, stick it inside the baby, inside the amniotic fluid, and take out some of the cells. And those cells are subjected to the sequencing. And there are lots of locuses where it can lead to different kinds of diseases. People sequence those locuses and find out if they have mutations or not. And that is a very common practice in general. It is slightly costlier business, but it's being done on regular basis. Yeah. You can imagine in India, there are a lot of people who do not have access to primary care. And, my frank expectations from all of you is that with time, you will be able to change some of the situation. Yeah.
Thank you, Rajesh.
Very helpful. Yeah. More questions? Okay. So,
Sumoy,
yes,
Sumoy has a question. She raised her hand, so I was waiting for her to say. Then I would ask mine after her.
Okay, I didn't understand. Over. Okay. So can you hear me? I don't know whether it shows that my internet connection. Okay. So I don't know whether the question is stupid or not, but I was just wondering that can stem cell be extracted from an aborted baby?
Yeah, it's a slightly it's a complicated issue. People have been trying to do that. There is only a certain stage from which you can extract. There is a lot more debate going on. Every scientist have some or other way of thinking about it. You might want to listen to John Gurdon, and he has a slightly different views than most of the modest, scientists. And John Gurdon thinks that if you know a technology and it just possible based on, if you allow it to be research, you as an individual should have an option of saying yes or no. So he says you should be allowed to do most of the research. And some of you in the future, whenever you become policymakers, will meet these complicated issues where you need to understand the soft part, which is, oh, it's a baby, and can you do this to a baby, no matter whether it's aborted or not, and really take out those cells. Some people say that, oh, it's an aborted baby. You can use the research and really help out the next generation of the babies and really save millions of babies. There is a heated debate going on at this point, but there is a very high limitation on what you can do, and it is limited at the funding agency. So funding agencies do not sanction projects like those, and hence the projects are not done, and there are institutional guidelines for work like those. Yeah, but you can do it in other animals, mouse and zebrafish. That's allowed.
Okay, okay.
Yeah, so
Yeah, yeah.
there is no rule against it. Yeah, I guess.
Thanks. Yeah.
Yeah, sure.
Yes, you got this question of like, is there a better way to detect sickle cell anemia in a baby before it is like born? Or so you said that you just extract certain cells from the embryo and so is it possible that, see, because from the parents 50%, like, chromosome comes from the mother, 50% from the father, right? So like, it's half half. So I mean, there are only certain permutation combinations that can be possible when it comes to like what is the genetic makeup of the baby. So there of course can be certain computational algorithms which can predict the genetic expression of the, because if you just do genetic testing of the karyotype of the mother or the father, then you can understand whether they are carriers of sickle cell or any other disease for that matter. And then I don't think that extracting cells from the embryo is necessary, because then if you know that they are carriers, then you can just come up with permutations and combinations of whether the baby can have a sickle cell or not.
So essentially, when it comes to patients like Manny Johnson and Aiden Johnson, who are brothers, they know from their family that they are, family has a sickle cell mutation, and hence there is a baby of certain chances, certain permutation, as you said, who will carry it. But eventually, even if you know the chances, you need to take out cells to be very sure, and there is no way other around it. And a lot of times, even when you know that the baby has a sickle cell disorder, aborting a fetus in early stages, it's a very difficult decision which a mother and father has to take. And there are only so many combinations can happen. And a lot of times people say, oh, the aborting a baby is not a good idea. Certain religion aspect comes in, and a lot of times you don't have sufficient money to do all of these things. A lot of times we have seen, at least in case of India, people are very poor. They cannot afford daily meal. So doing a test like this can be very difficult. But if some of you come up with a very easy way of doing it on a paper by just doing a chemical process, by having an enzyme which cuts only that region and leads to colorimetric test, and if it is around 20-30 rupees, and if you can design something like that, that will be a game changer in terms of how the analysis can be done. And many such similar inventions, not necessarily genetic testing, are actually done by this guy. His name is Manu Prakash, and he's one of my favorite scientists, and I look up to him. So you guys might want to check out his lab site. He's at Berkeley. Yeah. Okay. I'm gonna move on and take more questions at the end of this session. Is that fine with you guys?
Yeah, thank you, Rajesh.
Awesome. So, we just learned about taking stem cells and reintroducing beta globin corrected copy or BCL11A targeting shRNA or CRISPR-Cas9. But in this process, you need to take out the stem cells and put them back. And a lot of times when you do that process known as bone marrow transplantation, it's not 100% efficient. And lots of times these stem cells do not find a niche, and in that process, they die. To tackle this question, Zon Lab 10 years ago or so came up with this project, and they tried making use of zebrafish as a model organism. As shown here, the zebrafish stem cells for blood can be marked by RUNX1 and c-MAB in situ hybridization, which essentially binds to mRNA and gives you this blue colored reaction. During zebrafish development, their cells are born through iota and they are found in the CHT or the tail region of the zebrafish. This is the place where they will develop, they will multiply, and eventually mature and form a adult niche in the kidney marrow, which is very similar to bone marrow in case of vertebrates. They designed a chemical screen, and they screen for lots of compound which will increase the intensity of RUNX1 and c-MAB in situ hybridization corresponding to more number of stem cells. And they found out there are lots of molecules such as linoleic acid and prostaglandin E2 which can increase the number of stem cells. And here is the graph showing when looked for different markers for the stem cells, whenever you give prostaglandin E2, which is a small tiny molecule, the number of stem cells went up. Now, whenever it comes to a chemical screen like this, you need a male and female zebrafish. They lay hundreds of, 300s of eggs, and you can essentially put them in very, very tiny wells. And each of the tiny wells gets a very specific compound from the chemical library. And at the end of the development, you can look for a chemical hit, which gives you more number of stem cells as shown here in case of one of these zebrafish. Now, people have come up with different reporter lines, as we discussed, which actually mark stem cells in case of zebrafish. And one such a reporter is coded by the same gene which marks the stem cells as shown here, RUNX1. And we have a reporter in the stem cell in the zebrafish, which is a coding region for RUNX1, which is taken from a mouse, and it is still able to mark stem cells in case of zebrafish. So we have this reporter injected inside the zebrafish, and now you can see the stem cells going around in the body, and you can then add a chemical compound and really see the number of stem cells going up. And thus that allowed us to find this prostaglandin E2, which is now in the clinic when it comes to bone marrow transplantation. And this was a very important study when it comes to human use of zebrafish as a model to cure diseases and illnesses when it comes to human disorders. Now I'm going to play this video, and this is essentially animation describing what I described by Len, who's my current mentor. And we'll take questions at the end, but this is something which I found that was very exciting to see, that when it comes to stem cell birth and its process and finding its final niche, it goes through lots of different changes. And then lots of different niche cells also are very key when it comes to, just taking a stem cell and dividing it into, multiple stem cells and forming the whole blood. Okay, so I'm going to start this video.
To help understand how stem cells work in our patients receiving bone marrow transplants, we made a special zebrafish that allowed us to witness the birth of a blood stem cell in the body. The stem cell comes from a blood vessel cell. This blood vessel cell in green changes and rounds up and becomes a blood stem cell. At this point, it could be transplanted into a patient, just like we do a bone marrow transplant and rescue their immune system. This stem cell goes around and around in circulation and will ultimately colonize the next site where blood cells are forming, and that ends up in the tail. This stem cell has receptors on it, and it comes into contact with blood vessel cells that have the growth factor that binds to the receptor. This allows the stem cell to attach. Another growth factor comes and triggers a shape change, and the stem cell pokes through outside of the blood vessel. The stem cell will then travel into this space outside the blood vessel system, which is an environment or a niche. The stem cell is present and wants to attach to the gray cell, which is its nurse cell or stromal cell. The endothelial cells, or blood vessel cells, actually wrap themselves around the stem cell in a process we call cuddling, and this brings the stem cell next to the stromal cell. Divisions of the stem cells happens now, and the stem cells that are dividing out from the stromal cell will leave this pocket, whereas the stem cell that is attached will stay. This happens over and over again in many different stem cell niches. By 72 hours, there is a sea of stem cells that are cuddled by blood vessels, and they're sitting next to one of those nurse cells or stromal cells. By 96 hours, all of the stem cells actually leave, and they will go off into the blood vessel system and actually start to find another site of blood development, this time in the marrow. In the fish, this would be in the kidney. In humans, it would be in the bone marrow. Some of the cells actually bypass the kidney and ultimately go to the thymus. When they're in the thymus, they will start the immune system, and this allows the animal to have blood throughout its entire lifetime.
Okay. With that, I would like to thank you all for your attention, and my dear colleagues Nagraj and Vinay for this opportunity. And I would like to also thank my colleagues at Dana-Farber and Children's Hospital, whose work I have shown. And I'm immensely proud of what they have been doing on daily basis in hospitals and with patients. And I would like to also thank Lakshmi, Mittal and family for funding Scienspur and allowing all of us to be here and discuss this great science. Thank you. If you have more questions, let me know. I'll take questions now. Yeah.
Yeah,
Thanks,
Radha.
Rajesh.
Okay, you
As
say. Akhila, you say. No, say.
okay. As you said that it is possible to extract the stem cells from aborted baby, right? So when we extract such stem cells, does they retain the capacity to produce organs?
Yeah, that's a very good question. So, this whole field of taking out any tissue from aborted babies is a very it's a live debate at this point in terms of what you can do and what can be done, what cannot be done. Uh, but if you can possibly take out stem cells, let's say if you have to do the same thing with any other model organism, can you take a stem cell from a bone marrow of early developing animal? You can literally make the same the organ or the tissue from it. Now, people have been trying to understand how actually organ develops, and in that case, there is a hot research area known as organoid development. So that's something you might want to read about. And what they do in terms of organoid development is they take stem cells and all the other cells from the primary tissue. And in this case, you can imagine you really need material from early developmental stages or even when it comes to whole organ. And in that case, people have seen indeed, if you give enough amount of nutrients and the correct tissue media, those tiny amount of cells in a right combination can form a tiny organoid which can function as a lung, a tiny organoid which can function as a brain, and it's an very exciting area of research. One of my senior colleague at Harvard Stem Cell Institute, you might want to check out her lab. Her name is Paula Arlota, and they have been trying to do something similar for different kinds of organoids. Another colleague of mine here at the Children's Hospital is trying to do the same thing with the lung development. So her name is Carla Kim. And you might want to check out these labs and the work they have been doing. And it really flies close to what you just asked. So yes, in short, it is possible, but there are guidelines on what all you can do when it comes to humans and many vertebrates. Okay.
That means even if the organism died, the stem cells are alive.
Can you say that again? We are just breaking. Yeah.
Okay, then.
Can you repeat the
Even
question?
if the organism die,
Yes.
yeah,
So there is a certain
even
time.
if the organism die,
Yes. So what happens is when there is a death of an animal, there is a certain time after which cells will start dying. And the death of an animal, according to the standard definition, it's a death of the brain, which is considered as a death of brain or the stopping of heartbeat. It's considered as a signature of the death of an animal in general. But when that happens, the rest of the system is still functional till certain time, and that's why some people can donate some organs. So you can take out organ till certain time, and then really based on the permission, you can really do those kind of experiments and transplant them. Yeah, so it is possible.
Okay, thank you.
Yeah, Halak, why don't you ask question? You haven't asked any question. Manisha, hold on to your question for a minute. Yeah,
Sure, no
sorry.
problem.
Yeah,
Yeah, so am I audible?
yeah, you're audible. Yeah, you can be a little louder though.
Sorry, my voice. I'm little sick, so
Yeah.
okay. Okay, so I have a question that we have been talking about some niche and some triggers that can be used, and
Yes.
you've been talking about this chemical hit thing.
Yeah.
What chemical is specifically used, and what kind of situation that causes it? Like, hello.
So you're saying when it comes to a library of biological active compounds, how do the selection for which chemicals to begin with is made, and the chemical nature of these compounds? Is that the correct question?
Yeah, and how to know that with this chemical has been used, or what can the exact properties?
Yes. So what people have been doing, these compounds actually come from diverse sources, and the whole combination of compounds known as the chemical libraries, they come from companies, and these companies have actually done research about individual compounds in terms of their biological activity, their ability to kill a cell, and ability to modify different signaling pathways, and they have been researched till certain extent. Now, when you do a chemical screen, you start with thousands of compounds. You really don't know what exactly you're looking for. And then you find maybe out of 5,000 compounds, around 10 compounds which actually do certain things. For example, in this case, the number of stem cells going to niche is increased in comparison to the control. And once you find a compound like that, for example, in case of prostaglandin, what you do is you try and find the chemical properties of prostaglandin E2. Now, that information of what compound gave you the chemical hit in terms of your biology you're looking at comes from a company. But once you find out that prostaglandin E2 is working for your purpose, then you start modifying with the help of chemists that can you add some more moieties to this particular mother compound and make it more effective for the process you're looking at. And that is something which takes a lot of time. So it takes a lot of chemical biologists, it takes a lot of combination of tissues, a lot of experimentation using different dosages and different markers. And also, whenever you do an experiment like that, the first primary important property of this compound is that it should not be killing the animal. Okay, so and as simple as that, or it should not be carcinogenic. So whenever you put any chemical into market for clinical trials, you check for all of these. Does it lead into any other kind of abnormality in case of humans or in the model organism? And then it goes to clinical trial 2 and clinical trial 3. So it takes lots of different steps. Yeah.
Okay, but while taking this scenario, like sickle cell anemia may need newborn screening, right?
Yeah.
Yeah. So, but it newborn baby is not very adaptive to certain kind of treatments and chemicals. So, is it necessary to like? Is it okay to do with newborn screening, or what procedure has been held with the
Yeah. So
screening?
when it comes to any clinical screens, they are not done directly onto the patients. It starts with model organism, then you go on to do the same experiments using human cell lines, and then from the human cell lines you try and do those experiments with primary stem cells, which are non-cancerous stem cells. And when you show effectively in all the system that the drug works, then you go for clinical trials. And clinical trials are done on only few number of individuals with a consent, and then the experiments are done. So it takes lots of different kind of model organism. The reason why we use zebrafish, we have a zebrafish model for diamond blackfin anemia, and our lab found out that the calmolidin inhibitors, CAMK2 inhibitors, are actually something which elevate the symptoms of diamond blackfan anemia, which has RPS19 mutation, which codes for ribosomal gene. So yeah, you don't go for babies directly. Yeah, that can land you in trouble.
Thank you, sir.
Awesome, very good question. Yeah, Manisha, you can shoot now. Yeah, thanks
Yes.
for waiting.
No problem. I was just thinking that if we, in the video, we saw that the stem cells are functioning, they need the stromal cells for functioning. So if we have like a problematic stromal cell, then what will happen?
So Zon Lab is actually trying to study that in collaboration with Nancy Speck and one of the researcher who's very reputed when it comes to stromal cells and in general vascular cell biology, Shahin Rafi. And what we have been trying to understand using stromal cells and other kinds of sinusoid cells is, if a stem cell is fine, as you asked, and if the stromal cell has a problem when it comes to its property and the niche, will it lead to failure of stem cell and stemless? Yes, it is possible. And a colleague of mine, his name is Michael Fairchild and Tianxiao Han, they are trying to study different kinds of cells. So stromal cells are being studied by Michael Fairchild. And we do see that there are lots of different kinds of stromal cells when it comes to bone marrow niche. And this stromal cell research is something which is very complicated. The markers to identify what constitutes the stromal cells and the niche cell is something people are still trying to figure out. It's a very hot research. You might want to read about it. Yeah,
sure.
That's where the people are going. There is a cool technique. I just want to mention it, known as single cell RNA-seq. And RNA sequencing.
Oh, sequencing, okay.
Yeah, and what it does is actually, if you have a bunch of cells from the niche, you can take all the cells, and what it does in the process of processing the cells, it encapsulates into this bubble, which has all the enzymes to convert RNA into cDNA. And every cell in that bubble, tiny bubble, gets converted, every mRNA is converted to cDNA, and that cDNA gets a special barcode. So if you have 100 cells, every cell gets a badge.
Okay.
Now you take the whole cells, all the 100 cells, and sequence them in one go. And that is what gives you to know. Now you can know that every 100 cell is different or similar, or can you group them together? And I have been doing those experiments. I'll probably talk about that maybe in the fourth lecture.
Okay. Thank you so much.
Chamundeshwari?
Yes, Rajesh. Like now many countries are allowing to preserve stem cells, right?
Yeah.
So are they just preserve, or any experiments are being done on them?
Yeah, so what happens is whenever a newborn baby comes out, the umbilical cord, people thought that it was a great source of stem cells. So people have been storing it. But at the same time, you can just donate it for research. So, some of you can probably do that, or ask people to do that whenever, let's say you undergo an operation and a part of your tissue comes out, and they ask for your permission, can we take this tissue and use it for research? Yeah, I would say try and keep some of the rights with yourself, but help research. And one such an example I would like to tell is a very famous cell line known as a HeLa cell line. Henrietta Lacks was one of the African American who suffered from a cancer, and researchers were back then in the history were trying to grow cells in the dish, which is very common practice these days. But HeLa cells coming from this lady were the only cells which continued to divide because she had a mutation in her cells and they were cancerous. There's a huge debate about companies using those cells to do chemical screens and earn millions of dollars, and they're not giving any dollar to this family. Yeah, you might want to read about it. Yeah, so you can keep the stem cells and do more research, and that's how most of the research works. That somebody gives a tissue and it just happens to grow. There is a
I
researcher.
also
Yeah, go ahead.
no, Rajesh, like continue. I will ask
Yeah,
it.
there is a researcher here on Harvard University um, who donated his cells for the research, and his name is George. It's a very big research. He's the same person who's trying to bring back mammoth to life. He just got a big grant. Might want to read about it. Yeah. Okay, sorry. Go ahead.
Okay. My one more question is like many people will be suffering with heart problems.
Yeah.
Usually, heart transplantation is from life to life. That means you have to die, or we usually take from brain dead patients' hearts. So, my question is, if even these stem cells are used for research, so how many hearts does these stem cells produce? Is that they produce only one from a person extracted from one person?
So I think, as far as I understand, people are still trying to use a stem cell and really make a full heart out of it. And I think that has not happened yet. What you might have seen, there is a news making around. There's a research making rounds in news that this first person to receive a pig heart, and the way it goes is, as I described earlier in the last lecture, is you have a pig which is mutant for particular organ development, and then you introduce human cells into this um, pig, and the pig develops normally, and the only part which is possibly has only human cells because of the mutation particularly this pig has is heart. Then you can take that heart and put it in the patient, but you cannot really, till now, people have not been able to take a stem cell and make a full heart out of it. That is something which people are still trying to work out.
Okay, Rajesh, thank you. Can you like mention one more time that you told to read about George who donated?
Yeah, George Church. His name is George Church. He's one of my favorite scientists. George Church.
So how
Thank
is
you,
this
Rajesh.
mammoth cells even related with the human cells? Like, what is the fundamental concept behind that?
Which concept?
Yeah, like you said, he gave up for the research in mammoth cell, right?
Yeah. So what he is saying that if, let's say, you have a favorite pet, you have a dog, and you really like that dog, and then it was with you for 15 years, and now it's very ill and it's about to die, but you want exactly the same dog. Now the way to do that is to take a same one cell from the dog, take out its nucleus, and do really what John Gurdon was doing, which is putting that nucleus of your dog's cells into another dog oocyte and get the same dog, essentially the same dog, which is a cloning. Now, when it comes to mammoth, what has happened is, because of the years when it died, the number of nuclei which can be used for this and the amount of the genetic material, the quality of genetic material is compromised. So there is some fragments of mammoth DNA still intact, but there are lots of fragments which are destroyed because of it was just dead for so many millions, thousands of years. So the idea George Church has proposed is whatever is missing, we will take it from elephants, because elephants and mammoths are similar, and make this sandwich genome and then put that into elephant oocyte, and that will probably give rise to a mammoth. So that's the concept. And it's the same thing you can do to make a human being, if allowed, which will have all the super genes, nice muscles, blue eyes, blonde hair, tall, and whatever you can think of. So that's a concept that you can take a gene modify it, put it back, and make a baby which will have all the characteristics.
Thank you. I'm starting to looking forward to look and search for Tony's chart now.
Yeah,
Thank
George Church. You might want to search, read about it. All of you. He's very amazing scientist. Yeah, Sharwari, go ahead.
Yeah, hi. So I had a couple of questions. So,
Yes.
in the video, you talked about stem cell cuddling by the tissues and the cuddling part of it. So, is it a receptor like like mediated process, or because the cuddling must have certain like the tissues that surround the stem cell must have certain conformational changes in them, right? Like for example, in a membrane, like the lipids become unsaturated to get a kink so that the fluidity increases. So
Yeah.
like there must be certain kind of conformational changes, right, like in the tissue that's around the stem cells. So what are these changes, and also is it a possibility that they don't happen, or
Yeah. So
and
what
poor
lab has
cuddling?
been trying to do is trying to identify these receptors, and uh, the technology I just mentioned, which is single cell RNA-seq, has been very instrumental in deciding what all receptors are found when it comes to some of these cuddling endothelial cells. And the idea is to really dissect out, part of this tissue and subject it for the sequencing. And it's an active area of research. We yet do not know what exactly are the possible molecules which are expressed on the stem cells and on the niche cells, and that's what brings in the cuddling. There's a colleague of mine, his name is Sam, and he is trying to understand that in the context of very specific cell type and its interaction with the stem cells. Wiseman, who's at Stanford, uh, and collaborates with lots of labs in California for the regeneration. He has found out that and there is a molecule known as calreticulin, and which is involved in terms of stem cell interaction and deciding the fate. But it's an active area of research. Yeah, very good question.
Yeah, and also same question regarding this, that is there something called as poor cuddling, or is it just cuddling, or
Yes.
it does not happen? Like,
So there are different degrees of cuddling, and based on who receives cuddling, how the cuddling is received, the cells actually will react. This whole research comes from a senior colleague of mine who has his own lab, and I'll send you his papers. His name is Owen J. Tamplin, and you can read Tampling et al. Paper from Len Zon's lab, where I am actually currently now. And he has characterized that in great detail. He has also worked out some of the genes involved in cuddling process. So you might want
right.
to read it. Yeah.
Yeah, sure. And also, I asked you one question yesterday that out of the two stem cells which divide, which is let go of
Yeah.
the niche, and which is used up to basically make the red blood cell or whatever they are destined
Yeah.
to be made later. So, is that decision also depend on which of the two cells receive more stronger cuddling and which receives poorer
Yeah.
cuddling?
We do think that the way you can see even in this particular picture, when the cell is dividing, it divides at a certain angle, and then only one of the cell leaves faster than the other cell. And this angle distribution and other niche cells around those cuddling cells also decides which cell become more stem cell and goes to niche faster and finds out niche better than other cells. And all of these processes are very complicated processes. The orientation of cell division and the localized paracrine signaling are very important. Different interleukins are known to be involved. Wind signaling, Notch signaling is also very important when it comes to stem cell maintenance and differentiation. So it's an active process, active area of research. There are a lot of different things which are not known, and there's good research going on. So you might want to read more about it, and I can also
Right,
find out some literature for you guys if you.
right.
Yeah.
Yeah, please send it to me. And also, since you said that there is not much research on this, so is that so you talked about the gray molecule also being responsible for like the cuddling process and like attaching to. So can there be Also, a reason, can this also be a reason that the two of the cells which divide, the one which is closer to the gray molecule, because conformationally they divide in one pattern, right? So which one is closer to the gray molecule stays and which one is not, can
Yeah.
it go?
Yeah,
Is
so
that also possibility?
lot of these processes in the early stages, people do think that there is some guideline to it, but a lot of it is also stochastic. But
Right.
once the decision is made, it really happens within like 30 minutes, like fraction of minutes and seconds, and one thing leads to another. Like for example, notch and numb signaling. If you see the way cell divides, the moment one cell gets a high notch, in next 15 to 30 minutes, the other cell is already asked to differentiate. And that is something I'll be talking about in the next lecture. So stay tuned for it. Yeah.
Okay, great. And also, last question is that, how did they actually see this live inside an organism? Because this is an
Yeah.
animated video, but like there has to be like, for example, I in Anoop's lecture we talked about there being a way called pulse chase experiment or something like that, where you tag one molecule and you see wherever it goes throughout the cell, but then that is only for like, I guess, a unicellular organism or something like that. So
Yeah.
how would that be possible in a zebrafish, which is a multicellular
So
organism?
there are a lot of different microscopes available. And the first time when it was done using confocal microscopy. Confocal microscopy. And but having said that, there is a very cool microscope known as lattice microscope. Okay, everybody whoever is on the call now should go and Google for lattice microscope and look at the images. Yeah, I'll be talking about more microscopy in the next lecture. Yeah, stay tuned. Chamundeshwari, you had a question and the
Thank
question
you.
has been
Thank you,
answered.
Rajesh.
Yeah, no problem.
No, Rajesh, I forgot to lower my hand. Now I did.
But now you need to ask a question. Your hand was there. No issues. Don't worry. Yeah.
Yesterday you have sent a paper on nuclear programming in amphibians.
Yeah.
I just went through it and I have few questions. So based on that, there were two scientists who were working one group were working on one second, Rajesh.
Yeah. Are you the only person
one
who
were
read the
working on
paper?
xenopus.
Sorry, go ahead. Sorry to interrupt.
So one of the group were working on the xenopus species, and the other group were working on rana. So at a point, it was mentioned that like the xenopus, when the actual nuclear programming was done in xenopus, they were normal individuals, and where that in rana, the individuals they were not normal. So some kind of problem was raised, and it was not answered in that. So what is the reason?
So the experiment in nutshell is taking a nucleus from one species and then implanting that or doing a nuclear implantation into another species oocyte. Now you can understand there will be lots of different things which will be common, but at the same time, because of the species differences, there will be lots of genomic regions which will be very different. What happens when you take a nucleus from the same organism and put it in the same oocyte, the fit, the compatibility in terms of transition factor, histone, and other epigenetic modifiers is complete. And that's why you see a normal individuals to a certain efficiency. In this case, even when he says it was successful, the success frequency was 1.5%. Okay, so it's not 100%. It's only 1.5% of those embryos actually made into adult animals. But when you take it from another species such as rana and put it in the xenopus, the compatibility between the reprogramming factors and the nucleus is not 100%. And that is what causes early abnormalities in terms of development. So the nucleus, which was supposed to divide and form a lot of more cells, did not do a good job of dividing. And even when divided, it didn't take all the expression the way it's supposed to be when it comes to a normal nucleus, or same nucleus in the same oocyte. So that's a problem. Very good. Are you the only person who read the things which I sent? There's more people who are paying attention to that. Yashini, you have a question?
Yes, Rajesh. Actually, I was unlucky. I had some network issue and I got disconnected. And the last
Oh,
30 minutes I couldn't listen. But then I'll watch the recording and again, and I'll get back to you with those questions. But now I have two other different questions. Like when we transplant, like when stem cells are being transplanted to a model organism or to humans, when if it
yeah.
all is done, so what kind of protection will be given to the model organism or the humans if it is done? Like
Yeah.
the they undergo this immunosuppression as a
Yeah.
whole, or to whichever organ it is being transplanted, only that organ will be made immunoprotective, or how is it done, that
Yeah.
mechanism?
So the question is, if you have a stem cells from the same individual or a twin who essentially has same MHC haplotypes, then you don't need to give any immunosuppressant because the cells are essentially duplicate because of the the way the MHCs are on the cell. But when a donor is different and is not related, then the MHC molecules are different and they are sort of identified as a foreign, and then the host can attack the graft and known as a host versus graft rejection.
Mm.
Now, a lot of these processes have been dealt, and there will be a lot of immune suppressant which will be injected, and they are given for almost 6 months or so till the transplant is complete, and then they are slowly taken off the immunosuppressant. And there is a lot of different kinds of ways you can do it, but with time and genetic engineering, I think that problem will be solved. It's a big problem when it comes to bone marrow transplantation. Most of the grafts are rejected because of the host system finding those cells as foreign and rejecting them and killing those cells one by one. Yeah.
So can we do only for a specific organ, or the whole body has to go immunosuppressive?
So what happens is the immune cells are everywhere. You must have seen macrophages. They are just on search. You must see if there is a tiny wound in your body, the first cells to be there will be neutrophils. And once they go bonkers in terms of their immune activation, nothing can stop them. A lot of
Okay.
times, autoimmune disorders are because of that. Your immune cells turning against yourself, and there is no way of stopping them. Yeah, alopecia is one of the such a diseases disorders, which sadly takes away the hair follicles and leads to baldness, untimely baldness. Yeah. Yes, Sharwari.
Yeah, I have one more question.
Oh yeah, please shoot. Sorry, I thought you're done.
Yeah.
Yeah.
No, like if at all we try to treat a disease or a disorder with the stem cells, like if we derive stem cells from an affected person and we derive stem cells from an unaffected person,
Yeah.
like how far would these stem cells differ? Like will they have any difference in them, or there is no major difference in doing this therapy from whichever being the source? Like if at all a person is diabetic and we derive stem cells from the diabetic patient and stem cells from the non-diabetic patient, and which one, like what difference will it have?
Yeah, so that's a
From
very good question. It's a very, at the same time, very complicated to understand and answer. If you take a stem cell from a person who has a propensity to undergo diabetes and has seen diabetes, then there is a problem that the stem cells have also seen some kind of changes in the insulin pathway. Now, you guys will be surprised every time when we do experiment in terms of tissue culture, for the cells to survive, they have to be given externally the insulin. So every media has the insulin in it, and it is a crucial component. So yes, there will be some changes. How it will affect stemness is something which is debatable, and it's an exciting area of research. Yeah, very good
yeah,
question.
thank you.
Awesome.
Thank you, Rajesh. Yeah.
Sharwari, last question.
I have two, so second
Two
last.
last questions. Okay.
Yeah. So, my first question would be that if I take BC stem cells from a patient who suffers from sickle cell anemia,
Yep.
and if I transplant those stem cells into a person which is healthy and does not have sickle cell anemia, so basically I wanted to ask that the probability, so all of those stem cells won't get converted into RBCs that have sickle cell, like sickle conformation, right? So is it dependent on the genetic makeup of the stem cell itself, that it converts itself into RBCs, or it like sickle cell, or it does not? Or is it dependent on the fact that okay, the other cells around me are like, basically if it is transferred into a person with healthy RBCs, so will those cells then again make healthy RBCs, or is it like they will induce sickle cell inside that person as well?
So in case of sickle cell disorder, there is only one mutation which causes changes of glutamate to valine.
Valine.
And no matter where you take the stem cell, if it forms a red cell, the cell which will come out of it.
All right, great. And also, I wanted to ask you about these stem cells project that are carried out in the labs. Like for any lab to get funding, you need to provide certain hypotheses and certain um like a research proposal, basically, like this might be the outcome. So when you have no literature for it, like you don't know, like basically what, so how do you get funding for such, like now if I'm doing plant tissue culture for one organism, and I know that this for one plant species, so I can take like other plant species, and I this plant species had this outcome for this experiment, so maybe this will be similar for this plant species as well. But for stem cells where it's not researched a lot, so how do they come up with hypotheses and everything?
Yeah, so a lot of times grant writing is altogether different game, and a lot of times when we write grants to ask for funding, they usually have some preliminary data, and on the basis of preliminary data and the human mutations, you can say with ballpark estimate and statistical analysis, there is a possibility of this thing happening. Hypothesis, which is hypothesis number one, and then hypothesis number two, and then some preliminary data saying that this might be the reason why it should be happening. And a lot of times, granting agencies say that, okay, your hypothesis sounds good, and that's why the process is competitive. So you have lots of groups writing their own hypotheses, and then say maybe this is what is happening. They will say that maybe this is what is happening, and based on the grant the reviewer committee, they will decide which hypothesis suits well, and decide to fund the project. It is something which will require a lot of thinking, processing of information, and collaboration between different groups to come up with a viable hypothesis. And that takes years of time and months to write a good grant. I'm actually in the process of writing one such a grant. Probably if time permits, at the end of fourth lecture,